This is Chapter 9 from Gene Expression, from genes to proteins, where we look at transcription and translation. It basically starts about 100 years ago when Garrod was looking at diseases, including something called alkaptonuria. And alkaptonuria is a metabolic mutation which ends up producing black urine. Now this black urine is a compound called homogenistic acid, and it really comes from the result of a mutation in a metabolic pathway, and that there's a buildup of a compound in that metabolic pathway because of this mutation, and it manifests itself as this particular phenotype. Round about the same time, two scientists called Beadle and Tatum were looking at mutations in Neurospora, which is a fungus. And the fungus they were looking at, they induced different mutations, and they assayed specific enzyme activities, and they managed to find that each of these activities were due to a specific mutant gene. So, as it says, it proposed that genes control the synthesis of proteins, and that proteins are responsible for phenotype. So here we have the first indication that there's a link between genes, or your genotype, and phenotype, what you look like. So this chapter looks at the link between genes and proteins. Why would we want to bother with this? Well, because proteins do the work, and they make you, and your phenotype. And they're encoded by genes, which is your genotype. So if we change the genotype, we change the phenotype. Your DNA is encoded to messenger RNA. And the messenger RNA is read in a series of 3 nucleotides, and we call these sets of 3 nucleotides codons. Now, codons are therefore triplets, and one triplet of messenger RNA, one set of 3 bases, is specific for a particular amino acid. And there are 64 different possible codons. And now, why are they 64? Well, there are 3 bases in each codon, and so we have, for the first base we've got 4 possible choices, the second base we've got 4 possible choices, the third base we've got 4 possible choices. Multiply all those together, we end up with 64 possible codons. They've figured out this particular link. All the possible codons are here on this diagram, and we call this the genetic code. And you read this by the first base down here on the left hand side. If the first base is U, then you look at this particular row here. If the second base is, shall we say, C, then we look at this set of 4 bases, 4 amino acids here. And if the third base is, shall we say, A, then that gives us U, C, and A, which is coding for serine. We'll come across the genetic code more in later slides. This genetic code is also universal. It's the same, it's nearly the same in all living organisms. Therefore, whether you're an armadillo or a polar bear, you have the same genetic code, which is interesting potential, meaning that we can take genes from different organisms, transfer them into other organisms, and they will still work. The title of this chapter includes transcription and translation, and those are the 2 steps that are required to take DNA genotype to your protein phenotype. And these 2 steps were actually first coined by Francis Crick. It's called the central dogma, and his basic idea was, here's DNA. DNA goes to make messenger RNA, and messenger RNA then goes on to make protein. This first step is called transcription, the second step is called translation, and this being the central dogma. So on to the nitty-gritty. How do we make messenger RNA? Messenger RNA creation can be divided into 3 steps called initiation, elongation, and termination. Obviously, initiation is where we start. There The protein we're going to use to initiate, to start transcription is called RNA polymerase, right here. RNA polymerase is a combination of proteins, including one called TF2D, stands for transcription factor 2D, along with a bunch of other proteins. And they all bind a piece of DNA which we call the tartar box. The tartar box is at a place where we call -10. More about that in a minute. So once RNA polymerase, consisting of TF2D, binds the tartar box, it unwinds the double helix, and we start transcription at a place called +1. +1 then is 10 bases further downstream, or further 3' to the -10 area. Let's look a little bit again at this initiation. The RNA polymerase binds the promoter at a consensus sequence. A consensus sequence is one that is common between all kinds of examples of different genes. And if we look at this diagram, here we have a series of 7 different promoters, 7 different genes from E. coli. This area over here on the right, this is the +1. This is the start of the gene. The gene is here to the right-hand side. All this to the left-hand side is called the promoter. And about 10 bases back from this +1 site, at a -10 area, there is a set of sequences, and if we look at the common links between all these different sequences and all these different genes, we find that most of them have the sequence TATAAT, otherwise known as the tartar box. There are other consensus sequences further upstream. There's another one here at -35. We're not going to worry about that too much, because the tartar box is the most important one in this case. And so here's an overall picture of a gene. This yellow box here is the gene itself, and the green area over here is the promoter. And so the +1 will be at the beginning of this piece of the gene, the -10 will be in the promoter, and the RNA polymerase will bind at that -10. Of course, it's never that simple. There are 3 types of RNA polymerase. The RNA polymerase that we're most interested in is RNA polymerase II, and that is the protein that transcribes protein encoding genes, all the ones that make messenger RNA, and we will spend the vast majority of time looking at those. RNA pol I transcribes ribosomal RNA genes, and those are the ribosomal RNA molecules involved in making ribosomes. And RNA pol III makes transfer RNA, which is also used in translation, and those transfer RNA molecules convey amino acids to the ribosomes. A small diagram about RNA polymerase II. It is made of multiple proteins, as you can see in this diagram here. This little arrow on the gene here, that is the transcription start site. Transcription start site, TSS. And the arrow is like a little vertical line with an arrow on top of it, and that indicates the +1 region on a gene. And so all these proteins, including transcription factor 2D, which is this one here, that one there, they bind at the -10 region on this promoter and eventually create the entire RNA polymerase molecule. This diagram shows a series of DNA strands. There are 2 green strands at the top here. If you notice, the top strand is 5' to 3'. The opposite strand, being antiparallel, is 5' to 3' in the opposite direction. And they are complementary. As you can see, A goes against T, G goes against C. When RNA polymerase transcribes this region of DNA, it is going to use the bottom strand as a template. In other words, it's going to copy the bottom strand. And because it's complementary pairing, the strand that it makes when it's copying the bottom strand is going to be the same as the top strand. And so these two green molecules here are the DNA. This red molecule is the messenger RNA that is made from transcription of this piece of DNA. So if we see here, this first nucleotide is a C, and that's the same as the first nucleotide on the 5' strand of the DNA molecule. Here is the same diagram again, but without the nucleotides, and you can see the template strand is on the bottom, creating the messenger RNA, which is the same as the non-template strand. Got it? Good. One thing you have to do when you're initiating transcription is open up the DNA double helix. And here is the double helix on both sides, and here is an opened up region of the double helix. And this blue region down the bottom, that is our non-template strand, the one that is being copied by the RNA polymerase, which is right here. And this whole region is called a transcription bubble, because we've opened up the DNA double helix here and here, and we have transcription going on one strand. And this is a short diagram to remind you that new RNA, new nucleotides, anytime you're making RNA or DNA, new nucleotides are always added from a 5' to 3' direction in that way. So these are the new nucleotides coming in right here, matching up to their respective bases. Remember, there's a uracil matching up against adenine in RNA.